The overall goal of the following experiment is to derive cardiac progenies directly and exclusively from pluripotent human embryonic stem cells using small molecule induction. This is achieved by adding nicotinamide to undifferentiated human ES cells maintained under defined conditions. Next, cardiac differentiating cells are detached to form cardioblasts, floating cellular clusters, in a suspension culture. Once formed, cardioblasts are attached to cardiac differentiation media where they continue to develop into beating cardiomyocytes. Ultimately, results are assessed by immunofluorescence with deconvolution or confocal microscopy and by electrophysiological recordings.
The main advantage of this technique is that it allows efficient induction of pluripotent human embryonic stem cells exclusively towards a cardiac lineage by simple provision of small molecules, whereas in existing methods of multilineage differentiation, only a small fraction of cells pursue a cardiac phenotype.
There are a few steps to be careful with when preparing stock solutions. Take care to slow thaw Matrigel and human laminin from -80 degrees Celsius to 4 degrees Celsius overnight. Store media at 4 degrees Celsius and always warm up the media to 37 degrees Celsius before use. Also, when preparing human ES cell media, some of the supplements are added just before it is used. These include BFGF, ascorbic acid, human insulin, activin A, and transferrin. Next,